Hey, everybody. It's great to see. I think this is the first seminar of the term. It's great to have Hyun back with us. And I was just catching up. He got promoted, and he's an associate professor with tenure now in biostatistics. He came here to work with Consuelo back, 2009, and then spent a couple years there, and then entered the tenure track as an assistant professor, and has just been a force to be reckoned with in computational biostatistics ever since. He was the John Searle Assistant Professor of Biostatistics, which is fantastic. He's had a strong association with bioinformatics the whole time. We were involved in his recruitment. And what we're going to learn from him today is his more recent work on single-cell analysis, which is so important right now. In getting that right and having statistics that you can trust is absolutely critical. And there's no one around, frankly, that I can think of here at the University of Michigan that would be a better source of information than Hyun himself. So we're great to have you back, and thank you for kicking off our seminar series. You filled the auditorium, and we look forward to hearing from you. It's my great pleasure to have you be here with us. Thank you.
Thank you, Brian. It's a really great pleasure to talk about our recent work on a single-cell analysis. So today I'm going to talk about some computational method that is interesting in the sense that this enables some new types of single-cell analysis that wasn't possible before, but the method itself is just computational. So I was not working on single-cell study from the beginning, of course, and I was fascinated by the talk from Steve McCarroll, who came here about 2.5 years ago. And he was he started the single-cell talk with these slides. So this kind of slide. So I plagiarized him a little bit. So here, the bulk RNA sequencing we were doing is like we're studying smoothie. Okay. Now, when doing the single-cell analysis is like a salad because you can see every elements. So if you have two sets of salad, you know that this is tastes better versus this is not. Then you can examine what's in there, and you can figure out, for example, oh, this store uses gold kiwi and this store doesn't use gold kiwi. That's the difference. So that kind of analysis, you can do that when it's much easier and much more accurate when you have this individual cell result. And when you have this kind of smoothie-like mixture, a lot of different confounding vectors can be involved, and some misleading results can come in. So 2.5 years ago, there's two cell papers back to back published. Basically, this is a new single cell analysis techniques that allows barcoding droplets that contains the single cells. So because each droplet is barcoded, previous single cell technologies like a Fluidigm C1 has to prepare a library for each cell separately. So there might be cell-to-cell variation, the cost is high, and so on. But here, because we're preparing library per sample together, we can get rid of a cell-to-cell batch effect, and we can assay thousands of cells in a single library prep. And this is a high throughput, low per cell cost. So this is a great opportunity, and I was very excited to work with this. So the basic idea of this droplet-based single-cell RNA sequencing, including the 10x genomics and the dropseq, starts with either hydrogel or some droplets and the bead that actually have some barcodes. So basically, you are barcoding the droplets so that you know which droplet the read comes from. So and the cells are shooted in in some flow. So stochastically, some droplet will have one cell. Some of them may have two cells. And you do the in-droplet lysis and reverse transcription, and you can sequence each droplet, each RNA sequence read tagged by these cell barcodes. So you can demultiplex each cell. So each read by the cell origin, then you can aggregate the read coming from one barcode, and those are the profiles of the single cell expressions. So when the dropsheet came in, we contacted the Steve Mackerel's group, and we got the CAD file that they had this microfluidic device. We contacted this Dr. Hyun and the Yuchi Chen in the ECS department, and they are very the frontier in the single cell technology, but they also were interested in this kind of problem. So they fabricated the device, and Junhee Lee, we set up a dropsheet, the wet lab, so small equipment, and the Hojun Kwak, who did actually all the work, he's now at Cornell, actually did a lot of work to make the dropsheet happen in Ann Arbor just within a very short period of time. And since then, we have a lot of collaboration, including Mohamed Othman on the human retina sequencing, and with Gonzalo Abacus and Matthias Kreisler and Edgar Otto, who is doing the kidney single cell sequencing. So this was really great experience for me, but I don't have a particular disease or a particular scientific hypothesis I'd like to look into necessarily for this single cell study. The reason why I was fascinated is that this is the very future-looking method that will likely be persistent for a long time, and I just wanted to understand what the data looks like. Because so I'm going to plagiarize once more. So George Box said that all models are wrong, but some are useful. We can say for the technology too. So most of the high throughput technology, when you are getting, those are giving some useful information, some information, but those readouts are not perfect measurement of the real, for example, real RNA levels for each cell, for example. So when you look at the data, there are parts that are wrong or systematically biased, and correcting for that kind of bias or technical variability is what I'm very interested in, because making the analysis right is really important, and that requires careful statistics and computational analysis to achieve it. So I decided to work on a single cell method, and this initial experience gave me a lot of ideas about how to analyze the single cell data very well. Then about a year ago, my colleague, the PhD lab colleague, Jimmy Yang, and I talked about single cell technology because he was interested in it too. And we talked about it, and we realized that we have a very similar idea with each other. So this was the idea. So he was working on the 10x Chromium data. I was working on the dropsheet data, but I'll explain the 10x Chromium data first, because that was how the experiment was done actually. So 10x Chromium data is this platform is a very easy to use platform that can partition thousands or tens of thousands of cells in a very short short period of time, and it recovers 65% up to 65% of loaded cells. And if you load about 1,000 cells per well, you're going to have about 1% of doublets. So that's what it is. It's quite expensive. It's more expensive than dropsheet equipment you can set up. So it varies by how much you sequence, but at least per well, you're going to have to spend about $2,000 or more. So if you're doing the 8 well, it's going to be close to $20,000. So when we are doing the single cell analysis with this kind of platform, this is really good platform to create some atlas of certain single individual, single sample. And if you wanted to deeply analyze what kind of cells are there, cell types are there, and what are the profiles of each of the cell types, these are very good platform. But if you are interested in some kind of differential expression analysis, where you have a typical case control or 2 different conditions and try to compare the gene expression between the condition and try to identify what kind of genes are differentially expressed, you need some reasonably large amount of sample to do that analysis. Then, because you need a single well for each experiment, if you're doing the 10 sample, basically it's gonna cost more than $20,000. And for EQTL analysis, where you look at the association between the genetic variants and expression levels, in that case, you probably need about hundreds of samples to get a reasonable power. Then in that case, the study becomes more than $200,000, which is pretty challenging to accomplish. So second thing, so my point here is that when you do the wide experiment, it becomes more and more expensive because the per sample cost is almost fixed. So the second challenge is that there is a possible batch effect. So I think a lot of you know if you're doing the either experimental or analysis, but if you're careful to batch effect, you will always from microarray to RNA-seq and everything, you always have to randomize the covariates with the possible batch effect you're going to have in your data. Because, for example, if you do all the cases in one day in one well, one set of wells, and do the controls in the other day with the other sets of wells, these technical differences totally confounded by the biological differences, and it's very hard to disentangle which one is due to the technical effect and which one is due to the biological effect. So better thing is to do this match it assignment between the case and control. You can think about doing by well, but each well itself is also batch, so you still have some sort of batch effect, although batch effect is a somewhat randomized, that will reduce the power of your study. So ideally, if we can do something like this, so if we can put one case and one control in each well, that'll be very ideal setting to reduce batch effect, because in that case, you are perfectly matching between the case and control. So in this particular case, you don't need to worry about well-to-well batch effect or confounding your analysis. So this randomization is encouraged, but sometimes people come to us after doing the experiment, so it was totally confounded. There's nothing much we can do, and this kind of more ideal experiment was not possible with current protocol. So third challenge is that this is a stochastic procedure. So if you load more and more cells in the microfluidic device, there are more chance that the 2 or more cells come into a single droplet. So if you load more cells, you have to risk for having a larger number of doublets. Doublet means that the one droplet have 2 cells, or multiplet, which can have 2 or more cells. Then those doublets will look like a single cell, but have a very different expression profile. So it's going to increase your number of clusters, and it's going to give us weird systematic bias in your analysis unless you identify and detect them and throw them out. And detecting doublet is not an easy problem in general. So, because this is a pretty expensive experiment still, you might want to load as many cells as possible, because the speed and hydrogel, this library prep cost is very high. So if you reduce the number of cells per one, you're basically increasing per cell cost quite a bit. So there are many other challenges in the droplet barcoding single-cell RNA sequencing studies, and I only talked about 3, and there are other challenges I haven't talked about and that didn't address explicitly in my work. But I'm going to talk about a method that can handle these 3 challenges with some different way to design a study and analyze them. So let's think about doing the EQTL study. Let's say I need to have 96 samples to have enough power, and I'm assuming I'm going to have 1,000 cells per each to have a low doublet rate. So in this case, this is probably just experiment itself is at least a $200,000 experiment, could be even more. It could be cheaper later, but that's the price tag right now. So you can imagine, there's nothing wrong with imagining. If you can, so let's say I have only $40,000 to do the experiment. So let's say I have money for only do these 2 sets of wells, and wanting to do the 96 samples. If I somehow can load the 6,000 cells each from 6 samples together, that'll be really good. So this is a more efficient design for wide data. This will reduce the batch effect, because if you load the 6 samples together, at least within those 6 samples, you wouldn't have a batch effect, and you can even do like a Sudoku like a design where you put the sample in the 2 different combinations, and those approaches have been tried in DNA sequencing, and you can apply those kind of method here. So you can reduce the batch effect with a more collaborative design, and because you are sequencing 6,000 per cell rather than 1,000, you will have increased throughput. But obviously, this has 2 main problems. One is that if you just mix them together, how can you deconvolute cells to samples? So you somehow need to tag the sample somehow within a droplet, which is not possible now. And or you need to come up with some way to deconvolute cells to samples. And the second challenge is that how to handle the increased doublet, because if you load 6,000 cells together, you're going to have about 6% of doublet rates, which is very relatively high. So we came up with some crazy idea that just do that, and we're imagining if we have some magic box that can detect the doublets and the demultiplex the read by sample, then we can do this. Then how do we do this? The idea is that we're going to use the genetic variation that are carried by the mRNA levels, each of the mRNA reads. So this is a cartoon version of the basic idea. So let's say the genome sequence of some sample looks like this. And some of the RNA sequencing read carry some genetic variation. Here, it carries 3 genetic variation, and that matches to their actual genome sequences. But they are variable between the individuals, so you can use them as a natural genetic barcode to identify the sample from all the other samples. So then you can use this genetic variance information to deconvolute the read by each sample using this genetic variation as single cell barcode as a natural barcode information. So then if you, let's say you have 4 SNPs here, for example, 8 TGC, and there are a lot of different read coming from each represent a droplet. So then you have a lot of reads coming from here, and only this one matches exactly with this set of read. So if there's no sequencing errors, then just having these four different read from 4 different SNP should be enough to deconvolute the sample identity in this particular scenario. But obviously, this is not realistic because first, humans are diploid, so this one assumes haploid. This is a much easier problem. Second, sequence read actually have errors a lot of times sometimes, and the genotypes may not be accurate either. And you might have a very limited number of read that overlaps with SNPs because the SNP density is about one out of 1,000 or one out of few hundred at least. So we need to develop some more principled model that is a more statistically rigorous, and you can easily come up with a likelihood model like this. So this looks a little complicated, but I'll go over. So you basically multiply across all the variants, assuming they are independent, and go across all possible genotypes. And for each of the genotype, you're going across each of the read and calculate the probability of each read given the genotype, and multiply them and marginalize by the sequencing error, possible sequencing errors. Then this likelihood should represent the likelihood that this particular set of RNA sequencing read are coming from for some particular individual. Then if you have a list of the candidate individual you can evaluate from, then you can compare the likelihood and choose the one that gives the highest likelihood. And if you can give some buffer to decide whether this is ambiguous call or this is a confident call. So that's the basic idea that should work. We just don't know if this is going to be enough or this is not enough because of the lack of number of reads, or the model could be improved a little bit more. So more challenging problems are detecting doublets. So when you detect the doublets, the nature of problem becomes if there's two different individuals the 2 different cells are in there coming from 2 different individuals. Basically, you have 4 haplotypes rather than 2. So in the diploid model, you assume that each haploid is contributing 1-to-1 mixture, and you're assuming the tetraploid where 2 chromosomes are coming from with some proportion. Let's say alpha is like 30%, and the 70%, and this one comes like a 30% a mixture. If you know that what's the fraction of read that are contributed by each sample. So then, not every doublets can be detected, but if there's a doublet that are coming from the same individual, this genetic barcode itself cannot be used to detect the doublets. But if you multiplex a lot of samples together, most of the cell doublets actually should come from different individuals. So if you have n samples, let's say 10 samples, then 90% of the reads should be coming as 90% of doublets should consist of 2 different individuals. So you can if you can detect doublets this way, you can get rid of the 90% of doublets. So let's assume that this method works perfectly. then this is a theoretical upper bound of the performance. So if you can load more and more samples, then what's going to happen is that you're going to have more and more doublets. So you need to load in more and more cells. And this red line represents the number of non-doublets. So these are number of singlets. This is what we care about because we're going to throw out all the ones that are not singlets. Then our calculation shows that I want to have a 1% of doublet rates. Then for a typical way, you just load 1,000 cell and excluding 1% doublet, you're going to have a 990 singlets. But if you load 63 samples, each of them about 15,000 cells, the 1,500 cells, then you need to basically load 100,000 cells, and about 35% of them, 37% of them are going to be a singlet. So you're going to have less number of singlets per sample, but total number of singlets you're going to get increased by 37-fold. So this is huge increase, and this is obviously theoretical. We don't even know that the 100,000 read will fit into a single run because this is a huge amount of concentration of the cells. So this is a theoretical, so we need to see whether this actually works. To make it work, we need to develop the method to detect the doublets. It's basically the same method, except that you assume that there is two possible genotypes, and the likelihood of reading one particular allele is a mixture between coming from one sample versus coming from another sample with some mixing proportions. So this is actually developed several years ago. This is not new. This was developed a long time ago. So it was developed by us. So me and Mike Benke is not here, but Mike Benke and Gonzalo Elfects, we developed this method several years before because we knew that it's going to be an important problem in the single cell. So that's not true. So we did that because we needed this model. So we developed this model to detect the sample contamination in DNA sequencing reads. So this is a very similar problem indeed. So when you have a sample contamination from two different samples, the way to detect the contamination is develop this kind of likelihood model and do the maximum likelihood estimation to estimate the amount of contamination. The detailed setting is slightly different, but we are using exactly the same model. So we use this old model, apply this model, and implement the software and see how that works. So the setting we have, this is all done from the Jimmy Yes lab from UC San Francisco. And each sample is a genotype, in this case, about 250K to 500K GWAS arrays, and number of actually informative SNPs are less than 200,000. But it was imputed from the HRC panel, the Haplotype Reference Consortium panel. So we have imputed genotype, which is not correct, which is not always correct. And so the millions of genotypes that could be used as the genetic markers, but only about 1% of them are in the coding region. So we still have a limited number of genetic variants that can be used as a genetic barcode. So, and we had 8 samples and separated them into 2 batches. And we also had another well that contains all the samples together. And we wanted to see how they look like. So aggregating all cells together, this is the result of the clustering of cells. So this is a very typical plot for the single cell the community. So the x-axis is a TSNE dimension for first dimension and second dimension. These are 2 dimensions. So TSNE is called T-distributed stochastic neighbor embedding. which is a very good method to create a very low dimensional manifold from the very high dimensional data. So this is shown to work better than PCA or other kind of clustering methods. So this is used a lot to visually cluster the single cells, and we can use the known markers. In this case, these are all PBMCs. So to use known markers to classify the cells into B cells, T cells, and the different types of T cells. And dendritic cells and NK cells and so on. So this works very nice, and this is very consistent to what we would expect, except that this is actually a mixture from multiple different samples. So what happens if you demultiplex these samples? So this is what happens. So this is from well 1, which has only sample 1 to 4. This is from well 3, which has a sample 1 through sample 8. And we deconvoluted only the part that are assigned to sample 1. And this looks very similar to each other. And this is from sample 5. They look very similar to each other. But interestingly, these are not similar. These are not similar. The reason is that these are lupus patients, so they have a very different profiles of the cell type proportions. Also, when you sample the blood, sometimes you have more T cells than others, just by the variation based on by the people who draw the blood or who extracted actually the PBMCs. So they do have this very substantial differences in the cell types. So, and we wanted to evaluate how well this method do in terms of the accurately demultiplexing each cell. So the first well, we loaded about 4,000 cells, assuming that this could be deconvoluted very well. So we assigned each of the droplets into either confident singlets, confident doublets, or ambiguous case. So among the confident, so 97% of them are confident singlets. And among the singlets, so the way how we did that is that we just pretended we do not know that the sample are coming from only the half of the individual, and we try to we let our method Demuxlet to assign the best matching individuals, and only 0.03% of the case it assigned to the wrong half of the individuals, and most of the case it did assign the identity of sample into the right clusters. This reassures that this method actually do multiplexing pretty accurately. In well 2, we loaded a little more cells, 4,200 cells, and in this case the numbers are very similar. So 96.6% singlets, and they were assigned correctly to the right half in a 99.99%. And in the well 3, we used all the A samples. In this case, we don't have this measure of the accuracy, but the number of singlets are decreased because we loaded more cells, which is of 6,000 cells. So we expected that, and this is very in line with what we expect based on the estimated doublet rates. Also, we can view by the proportion of cells in well 3 and well 1 and well 2 by each individual and each cell type, and they look very concordant. So this looks like very reproducible result, even though we use the multiplexing the techniques. So we plotted this doublet race to the previous published TEDx paper by Zhang et al. In the Nature Communications, and this is our doublet rates estimated, and these are very in line with what others have done with experimentally, but the the people have done usually using the human and mouse mixtures, and they look very concordant. So there's another application of this method, which is quite interesting. So now these 8 lupus patients, blood PBMCs are extracted, and we put the interferon beta, which is a potential drug target, and let the sample stay for 6 hours, where the interferon beta effect could be maximized, and measured how the profiles will be different across the cell, across the different conditions, also across the different cell types. So this is a view between the conditions. So this is all T cells, mostly like T cells, NK cells, and these have a very different profiles because this interferon beta changes the immune response of the cells quite a bit. So expression profile changes a lot. And so this is known, and this is very cool view by showing that most of the cell types actually do undergo very significant amount of changes. And still, when we use the no markers to classify each cell type, it does show that these are all the same cell types. This is a great result. So in this case, we even loaded more adventurously. So we loaded 15,000 cells because now we know that this method works, and we know that loading more samples are going to be more advantageous as long as our method is close to perfect. So this is a plot I like a lot. So basically, each column represents different individuals, and these are 2 different conditions, and these are the different color represent the different the proportion of each cell types. Then what we did is that we asked the question that whether is this gene more variable between the individual than you would expect. Okay, so then if you do the test that you would do for bulk RNA sequencing just by summing up everything, you get this light blue or pink plots. That means that most of the genes are differentially expressed between the individuals. So those are highly variable than you'd expect. But that actually are caused by the different cell type proportion. So if you match the cell type proportion and try to measure the same thing, you don't have that many many genes that are differentially expressed by different individuals. So this is a really important result when you do the differential expressions. Always there is a risk of having the cell type having these different cell type proportion between the cases and control, for example, confound your analysis that most of the differential expressed genes are just specific to one cell type versus others. So that doesn't mean much in terms of the disease etiology. So this result is really encouraging that you can actually more precisely understand what's the gene that are actually differentially expressed within the same cell types, which would be more interesting. And you can disentangle the effect from the cell type proportion and the actual differential expression result. Okay, so we did the EQTL study too, and we used only 2 wells. So as you expect, so 23 individual, 2 batches, one from 8 sample, one from 15 samples. So 23 samples is a pretty large number of individuals. So it's not powerful enough to detect a lot of EQTLs, but we can at least with these samples that we can validate the existing EQTL or detect a very, very strong EQTL. So here 23 samples identify only the small number of EQTLs here. But interestingly, these are all colors represent the different cell types, and you do see that the EQTL profiles are slightly different by the cell types. And there are cell type specific EQTLs. The way we checked is that the green is the Jovardis LCL EQTLs, which is enriched across all the cell types. But also there is a cell type specific EQTL that was done by looking at the CD4 positive T alpha cells and CD14 positive. This was, I think it was an NK cell. Then CD4 positive ones, this is a T helper cell, so this is more enriched here versus here. And for this one, this is a opposite. So this is a, sorry, this is not the, this was not the NK cell. This is a cytotoxic, the mononuclear cells, and these orange ones are more enriched here versus here. So this is in line with known cell type specific EQTL as well. And also these cell type specific EQTLs, if you look at the actually distribution of the expression levels by the genotypes, you do see clear differences here. For example, this is ERAP2, which should be shared across most of the cell types, are just showing the similar patterns. But here you do see in some of the cell types, the effect is not observable at all, and only the subset of cell types you can observe the effect. Here, the same thing happens here for these T helper cells. You don't see the effect, but here in the mononuclear cells, or here actually NK cells, but this one is NK cell specific ones. So you do see a very huge effect by the genotype. So this is a gene environmental interaction or cell type specific EQTLs. So because we did a more adventurous experiment, we now plotted them against the known ratio doublets, and these are also in line. So you might think that 30% is really, really bad, but actually we are removing most of the doublets. These are 15 samples together. So then these are 26,000 cells. About 30% of them are doublets. You still have almost 20,000 singlets you can obtain, as opposed to 1,000 cells you can get if you didn't do the multiplexing with a similar amount of doublets. And so this lines up well with the projected estimates, and we are very happy with this result. So that's where we stand in terms of development, and we have a lot of different ideas to how to extend this method to make it more accurate and to be useful in different set of settings. And the topic itself gives us a lot of ideas, and it was really great to have that, have this going with a very good colleagues. So the summary is that we developed the Demuxlet method that demultiplexing samples and do the doublet detection using Lycra model. And our method basically enables cost-effective study that are genetically between the genetically diverse sample for differential expression and the EQTL analysis, or we call these per individual profiles of this TSNA plot as a drop print, because it looks like a fingerprint, and it actually is individual specific. So you can do these kind of census, and droplet rates goes as expected, and all the singular assignment we've seen that most of the cases 99.5% or greater singular assignment correctness, even if we multiplex a lot of samples. And yeah, so the benefits, as I said earlier, you can do wide experiment. So per sample cost is reduced a lot, and you can reduce batch effect, and you can reduce the per cell cost as well, because you can load a lot of cells. So the library prep cost goes down a lot by cell. So with that, I'd like to close with the acknowledgement of my lab colleague, Jimmy Ye. He's at UCSF, and he was so we discussed same idea together, and he had a very good plan to use this method to understand the genetics of the immune cells, and that worked out really great. And the papers out, the bioarchive, so you can look at what the details of the result. And we're very excited to work on this problem with in collaboration with Jimmy's lab. So that's all I have, and thank you.
Sure, we have time for questions.
Yeah. Well,
When you do when you're trying to assign the reads that you get from the single cell to an individual, you've got a small amount of reads that you're misassigning, or a small amount of cells, I guess, that you're misassigning. In the studies that you showed, you've only got a few other people in there to whom you could assign the reads to. So you've got 4 other people or 7 other people. If you go to having 100 other people or 1,000 other people, do you expect that those levels are going to go up, or is there something just not good about the sequencing of those particular cells and those rates will stay constant?
it's a very, very excellent question. We actually did some calculation of how many read or unique read or number of genes in terms of per cell, the quantity of the number of informative mRNA transcript will be needed to deconvolute from this particular number of samples. For example, if we have I just wanted to pull up these numbers.
Yeah.
So if we multiplex 4 samples together, then we get this accuracy. So we think we can improve the accuracy even more, but the current numbers suggest that to obtain more than 99% of the single assignment accuracy, from the 64 samples, we need about 50 reads that overlaps with the SNPs. So read from at least 50 different samples. 50 different SNPs. So that gives some idea. And basically, if you use this method with a lot more individual with a lot less read, you might have to throw out some of the read because that's too ambiguous. But if you deeply sequence the droplet well enough, you usually should be able to get at least a 50 SNP overlapping read because you still have a lot of reads, and that's a problem related to the capture efficiency as well. When you do the primary cell, you have a capture efficiency problem, so you usually get much lower number of reads than you would expect, and this is actually from PBMC, so that this shares this kind of problem too. So I think this actually works in the primary cell.
Thanks.
I have a question regarding the doublets. Why you're throwing away the data from the doublets? If you deconvolute with the genetic barcode, with the fingerprint, you might be able to look at the doublets as they were single cells as well, because you have both data sets.
Yeah, so that's excellent question too. So yes, you're definitely right. So you can look at the doublets and try to understand what kind of mixture of cell type that are in. So you need to know the cell type for both of the contributing cells, and also you can use the read information to deconvolute which cells are coming from which. So I think that's an interesting area. I think that's harder problem to solve because you have a limited number of information, and now you're solving the problem with tetraploid rather than diploid. So in general, you're solving the problem between the 2 sample mixture and try to get the right number of per cell quantification appears to be harder, and even just finding out what's the pairs of the cell type they're contributing from is not. It's a doable problem if you have a biomarkers, but if you don't have the known biomarkers, it's actually a hard problem. Yeah, so we have some ideas about how to solve that problem. Basically now, the nice thing about this data is actually, you know, you can just ignore the genetic information, just use the read count data and try to pretend that this is actually, now you know which one is doublet and which one is not doublet, right? So you can use this data as a reference data to try to evaluate whether you are really solving that problem. And there are actually, I think the human cell atlas groups are actually working on that kind of problem. But basically, to be able to do that, you need to come up with a mixture model that models the per cell type expression levels well, and try to model them as a mixture between two cell types versus single cell types. This is an interesting problem. I found that that's more challenging than I thought. I thought that simple EMI would work, but it doesn't work as well because of there are a lot more parameters you need to estimate. It doesn't converge well, so we're working on that problem very actively. And I believe that using the genetic markers and the expression level together should be the best way to more accurately deconvolute the sample identity as well as the cell types.
You talked about cost.
Yeah.
Just in terms of actual cost, how much would you experiment that?
It really depends on the sequencing, how much sequencing cost will be, because this doesn't necessarily reduce the per sample, per cell sequencing cost if you wanted to go deeper for each cell. In terms of library prep, it's really just 1 divided by, number of factors you're going to overload, because you're going to use the same number of hydrogel to load, and you're just increasing the cell flow rate. And one advantage I didn't mention clearly is that if you have a very valuable cells that you don't want to lose much, if you use the traditional approach, you will lose a lot of cells stochastically. But if you increase the cell loading, you will minimize the cell loss, and you have to filter out the doublets, but still you will save a lot more singlets using this approach. So this is a library prep cost. I think 10x says that it's a $2,000 per well. If you go by list price, basically you're saving 2,000 times a number of sample minus 1, I think. So yeah, for each well. Yeah. Yeah. So as I said, it's in 2 different settings, doing the 96 samples versus this way. It was about 6-fold and 7-fold differences just for the library cost itself. So the cost increase could be, the cost reduction could be the more than order of magnitude, so more than 10 times in a particular situation.
Yes, here, thank you. Can you imagine this approach being extended to chromatin accessibility approaches? What would be the challenges?
Yeah,
and
I
the
believe so.
opportunities?
Chromatin accessibility, you have a more diverse range of the reads. So usually chromatin accessibility case more, it's because there's so many coverage. Usually the number of read per cells has to be larger, so you will have the higher chance to get the variant overlapping reads, and that all can be used to deconvolute the samples. So I believe that our method can be used, and actually we don't necessarily restrict our method to work only for the RNA sequencing data. So any kind of sequencing data that are multiplexed by sample with a specific barcode tag, you can use it. So I believe you can, if you have that kind of data, you can use it. Thank you.